really interesting familial syndrome of Fanconi's anemia, which is near and dear to my heart. And I am very excited to hear what he has to say, since I'm not sure I've ever heard you talk about Fanconi's, Gary.
Thanks, Steve. So 30 minutes is always a challenging talk, so I'll do my best to get through what I want to talk to you about. But I know, it's perfect. But, you know, how's that? Okay, closer. So this is the quintessential genetic model of cancer, which is really what I want to tell you about. And not so much that I'm going to make everybody a Fanconi expert, but hopefully convince you that not only is the science really cool, but also that this is a disease that really has importance for everybody in the room whose career touches both pediatric and adult oncology. And so hopefully that'll become clear as we move on. But I don't have to convince you, though, that genetic syndromes, cancer models are rife within oncology. This is the APC colon cancer model, which a definable set of genetic alterations results in full-blown cancer. And Fanconi is part of that list, and this is actually a subset of that list of cancer susceptibility syndromes. The inherited bone marrow failure syndromes, of which Fanconi is one of them, and this is kind of the hall of fame of those various syndromes. But Fanconi, even though it's up until recent years has been kind of in a dusty corner of science, is actually the most common inherited bone marrow failure syndrome. And the other common thread here is that these inherited bone marrow failure syndromes are actually cancer syndromes. And although rare, Fanconi being the most common of these rare syndromes, they're incredible genetic models for understanding science. And then how can we adapt that science to move over to perhaps some therapeutic lessons? So I hope we'll get into some of that today. First, about a little bit of a primer, not only for Steve, but for all of you. Fanconi is an autosomal recessive disease, except for one X-linked variety marked by birth defects, many of which I think will become more familiar to those of you who went through med school and saw some of the classic pictures. Bone marrow failure or aplastic anemia, and this peculiar hypersensitivity to DNA damage, alkylating agents specifically, with associated genomic instability and leukemia and solid tumor susceptibility. So these patients actually look syndromic. They actually look unique. If you put them all in a room at summer camp, you'll actually notice that they all kind of look alike, even though it's not all that apparent to you in this picture. The other interesting, fascinating part about these patients is they have thumb abnormalities. They could be having this kind of aberrant looking thumb or be missing their thumb altogether. And on a radiograph, an X-ray, they may be missing their radius, which is in line developmentally with the thumb. And just to preempt a question later on as to why that's the case, I have no earthly idea, no one else does either. But there's this relationship of development with radius and thumb, which is part of this genetic syndrome. From our vantage point, it's this transition from a normal healthy bone marrow to bone marrow failure over here, and then with this ominous progression to leukemia, which concerns us the most. And this is a true cancer susceptibility syndrome. Now, I mentioned to you that these patients have a hypersensitivity to DNA damage. This really came out in the birth of chemotherapy when these patients were treated for their leukemias or solid tumors. And it's this real specific type of lesion, the DNA crosslink, which can be engendered by plain old chemotherapy. But actually, if we stop for a second and think about it, we're awash in DNA damaging agents all the time, whether we're on the airplane, cosmic radiation, we're breathing oxygen, or exposure to aldehydes, which are part of normal metabolism. These are all agents which will engender various types of DNA damage, but it's really the DNA crosslink which is the chief offending agent. Now, if you have a little kid coming into your clinic with pancytopenia, you'll actually, even if they look completely normal, because what I should tell you that a Fanconi patient can look completely normal. We always will suspect and test for Fanconi anemia in the case of a patient with pancytopenia. And this is a test you'll actually order right in Epic. You'll order a chromosome damage breakage assay, and you'll actually expose metaphase arrested cells to diapoxybutane, or it could be mitomycin C, or it could be cisplatin, any agent that will cross-link DNA. And you'll get a definable number of these broken chromosomes or stuck-on chromosomes or radial chromosomes. You can actually count that up, and that's actually diagnostically and quantitatively a test for Fanconi. The other way you could do it, however, is take cells from those patients, expose them to the same agents versus a wild-type control, or these mutant cells corrected with the wild-type cDNA, whichever variety that you want. And you can actually show a definable, statistically significant hypersensitivity to all of these different agents versus your wild-type control. You'll see this little assay over and over again. It's very simple, but very elegant, because this is really part of the gold standard for how patient cells, how cells that are deficient for Fanconi anemia genes have to behave. So there are 22, at least 22 different genes, any one of which is mutant biallelically or on one allele if it's X-linked variety, FANC-B. But any defects in any of these genes will lead to Fanconi anemia. I'm not going to go through all of them in any detail, but just to point out a couple of salient features. First of all, there's at least 8 of them which coalesce into a core complex of proteins with E3 ligase activity, which I'll come back to momentarily. The other thing is that many of them are actually breast cancer genes, and it was the cloning of BRCA2 that once it was appreciated that the FANCD1 group is actually BRCA2, which really brought this disease out of sort of the long forgotten neurons of everyone working in DNA damage repair and into mainstream cancer biology, because it was clear that these proteins are part and parcel of homologous recombination. So, and we'll get back to all those concepts in a minute, but just to sum up, this is, yes, a bone marrow failure syndrome culminating in leukemia risk, but these patients, if they're lucky enough to grow up to adulthood, get solid tumors, a lot of the head and neck of the genitourinary tract as well. And so what the current state therapy is, you transplant these patients, they grow up, and then they're getting a whole host of solid tumors that we have to worry about. And marked biologically by this DNA damage hypersensitivity phenotype. So this is the biology kind of summed up in one slide, and although I'm going to make you sit through another few minutes of biology, 22 different protein products which have a core complex which puts a ubiquitin on the central complex called ID, which then brings about a whole bunch of other proteins into play. And you see here by immunofluorescence after DNA damage, this diffuse staining becomes punctate. This is a FANCD2, one of these proteins here. And this is what a lot of DNA repair proteins do. They respond to damage, they come together into repair foci, and then they pull together other proteins. And that's really what's going on here. And you can see this is a hall of fame of DNA repair listed here. RAD51, BRCA2, BRCA1. And it's turned out that all of these are actually bona fide Fanconi genes. So just going to give you 3 little research vignettes that are going on in the lab to give you a flavor for both what's going on the basic science side of what we do, and then how we're trying to pull things over to a more therapeutic applied project that kind of takes some of the biologic lessons that we learn, how can we adapt them. Case number 1, this is an overview of homology-directed DNA repair, and this is actually work we did with Patrick Sung, have been ongoing with Patrick Sung, that focuses on BRCA1, BARD1. And BRCA1, BARD1, proteins which are intimately involved with homologous recombination, a coordinated set of steps which which go from DNA lesion to recruitment of the homologous chromosome, invasion of that chromosome, read off of that chromosome, and restoration of a normal DNA helix. And it's really formation of the central DNA repair intermediate, the D loop, which is formed by the invasion of that homologous chromosome, which is the hallmark of this process. So BRCA1 BARD1, probably don't need to tell anybody here what this is all about. BRCA1 being the first described breast cancer gene, but little did any of you in the room know before today that BRCA1 is a bona fide Fanconi gene. And so this is what really pulled, along with BRCA2, pulled the whole field into mainstream cancer biology. And again, not to overload you in 30 minutes with all the intricacies of what we do in the lab, this is merely, This is just really to show you a little bit about what we can do. We can actually make these proteins in vitro. We can make BRCA1 BARD1 in millimolar amounts. We can actually, in a coordinated, in a coordinated fashion, we can incubate radioactively labeled DNA with RAD51, the homologous recombination enzyme, along with the BRCA complex. And you can actually reconstitute these homologous recombination intermediates in vitro, as evidenced by this slower mobility D loop on a gel. And you can see that by adding RAD51, BRCA1, BARD1, you can actually create these D loops as measured here, all in vitro. And by doing that, you can then reconstitute a whole model, and you can go all the way from double strand break all the way to restored double helix, and demonstrate that BRCA1 BARD1 is involved in multiple levels of these, including the processing of the double strand break, the loading of RAD51, the recruitment of the homologous chromosome, and eventual restoration of the double helix. So another little vignette. This is actually taking clinical data and then going back to basic biology. This was actually data that I found out about just by going to the Precision Medicine Tumor Board here at Yale. And this was actually a head and neck case of an adult who had 2 mutations in the FANCD2, in the FANCD2 gene, one being a germline point mutation. And then this patient had a acquired deletion, 6 amino acids in FANCD2 in a conserved region of the protein. So we decided to work this up a little more, and we actually did a mass spectroscopy experiment of FANCD2, either wild type or missing that domain, and found a whole bunch of different proteins that were coming down in that particular domain. And one in particular caught our eye, and that was the Bloom's syndrome helicase. So Bloom's, which hopefully many of you have heard of, is Bloom is a RECQ helicase. It's actually very much involved with homologous recombination. Defects in Bloom's helicase results in chromosome aberrations. And this is another true cancer susceptibility syndrome. So we actually further mapped this little region that was missing within that particular case of head and neck cancer. And we actually were able to identify one particular residue, the 822 leucine, which when mutated to alanine resulted in the abrogation of interaction with FANCD2. So in this situation, you see FANCD2 being pulled down by immunoprecipitation here, Bloom syndrome protein being pulled down here, except in the case of this point mutant. So by all accounts, this seems to be functionally a very important domain. We can further show that this is functionally important by expressing this particular FANCD2 mutant in mutant cells. So we have these baseline mutant cells, which I'll remind you again are hypersensitive to DNA damage. If we provide the wild type cDNA back, these cells are thus restored for wild type resistance to mitomycin C. But if we express this point mutant of FANCD2, there's still, the cells remain hypersensitive to DNA crosslinkers. We can actually see the phenotype biochemically. As I mentioned to you, FANCD2 becomes ubiquitinated by, upon DNA damage. As you can see here in the wild type lane, in either of this 561K to R mutant or the 822 mutant, the ubiquitination event is actually diminished. So biochemically, as well as from a cell biology point of view, we can actually prove that this patient tumor-derived DNA sequence information is actually functionally important. So we've actually done this on numerous occasions to try to actually show that these mutations derived from clinical cases are actually functionally important. Then finally, we can actually show that ID2, the ID2 complex composed of Fanci and FANCD2 actually has functional importance. So we know that bloom can actually cause the dissolution or the unwinding of a double-strand substrate, and you see double strand goes down to single strand here. By addition of recombinant ID2 protein, you can see virtual complete inhibition of that process. And that point mutant, without showing you all the data here, the point mutant we showed to be functionally important in the previous slides actually is completely non-functional in this assay. So by biochemical and cell biology assays, we're able to show that this is a particular important region of the FANCD2 protein, both clinically as well as biologically in these assays. So it appears that bloom has, we know that bloom has function in the processing of the replication fork. And so the replication fork encounters damage and it stalls. And then a whole bunch of other proteins are recruited because there has to be protection of this replication fork during the process of repair. And so in the wild type situation, we have BRCA1, BRCA2, and RAD51 protecting this fork with the addition of ID2 and bloom. But when bloom is there alone, this all unwinds in an unregulated fashion, and nucleases come in and degrade it. So when you have proper protection, proper modulation of bloom helicase, you're able to protect that replication fork. So this is not the only scenario in which the Fanconi pathway intersects with other DNA repair pathways. Bloom being this particular example, we also have work we've done in mismatch repair proteins interacting with FANCD2. And actually, other DNA repair proteins like XPF are actually bona fide Fanconi genes as well. So it's clear that Fanconi anemia genes don't act in isolation. And there's a whole whole web of network of DNA repair pathways that come together to repair complex DNA lesions like a DNA crosslink. So this is kind of an overall view of what actually goes on in my lab, but this is sort of an overall view of what happens during DNA repair, which really can be thought of as a signal transduction event, initial processing of a DNA lesion through nucleotide excision repair, and then activation of the central Fanconi proteins, D2, Fanci, to coordinate downstream processes such as replication and repair, and then transcription, a whole nother set of projects which I don't have time to tell you about today. And then finally, how can we kind of take these biology lessons and apply them to therapy? And this is something we're very much interested in for a few reasons. First of all, for all the world, Fanconi anemia looks like myelodysplasia, and in fact acts like myelodysplasia. Fanconi is a pre-leukemia syndrome. And it may not be all that, it may not be completely empirical that that's the case, because we know that in the literature, there are actually FANC-L, one subgroup of Fanconi, FANC-L mutations which have showed up in MDS cases. We've also found a connection in work I'm not going to tell you about today between splicing factors, which are known to be multiple ones, mutant in MDS, and Fanconi proteins in terms of interaction. In sporadic cancers, there's actually a wide range of cancers which have been reported with sporadic Fanconi gene mutations. Jeff Sklar actually looked at this for us and had noted about 15 to 20% of all deposited sequence in COSMIC has Fanconi gene mutations. Now, with the caveat, of course, that we don't have specific data that all of those mutations are functional. Have functional impact, except for the ones that we've actually looked at directly. But These are certainly, this is certainly a sizable percentage for a rare genetic syndrome. And just to remind you, these are not patients with Fanconi anemia. And then finally, BRCA mutant tumors, which, remembering again that BRCA biallelic, if you're biallelically mutant for BRCA genes, you have Fanconi anemia by definition. BRCA2, BRCA1, part of the biology of FA, and of course, PARP inhibitors are an important and new part of the cancer therapy armamentarium. So we wanted to look a little bit more broadly at the scope of Fanconi biology and how that could be applied across this broad swath of tumors that are potentially mutant in Fanconi genes and defective biology. So we actually took a genetic screen using an siRNA library with the goal of, could we eventually identify pathways which, when targeted, could treat non-Fanconi patients with Fanconi gene tumors, mutant tumors, or Fanconi patients with head and neck tumors, for example, who really have very few options. It's one thing to do a bone marrow transplant preemptively and prevent leukemia, but there's nothing you can do to prevent a Fanconi patient from getting a head and neck cancer as an adult. And so the idea here is to find a knockdown of a genetic pathway which causes wild type to have much greater viability over the mutant. And that's really the definition of synthetic lethality. I won't go much into the suppressor screen, except just to show you how we set it up. And we did this at the West Campus in a high throughput fashion and got a relatively limited number of interesting hits, including PLK1, WEE1, which in particular are linked because WEE1 is a substrate of PLK1, and they actually have inhibitors in clinical trials that you can take right off the shelf, and we've actually used in the lab. And in particular, and this was done in a D2 mutant background, PARP did not show up as a hit. And so I think the lesson here, and we've been able to show this repeatedly in the lab, just because you work on a DNA repair pathway doesn't mean that those particular cells, when mutant, are going to be sensitive to PARP inhibitors. PARP inhibitors work clearly in a BRCA mutant background, but not in other Fanconi gene mutant backgrounds. So I think that was a lesson that we've learned. And just to show the data on WEE1, when we knock it down, we can show that there's significantly lower viability in our mutant cells versus wild type cells. And in this case, we actually functionally evaluated another mutant that we obtained from the Precision Medicine Tumor Board in another head and neck cancer case, this being a mutation of FANK-A. And here you have wild type FANK-A or that mutant version expressed here. Ubiquitination of FANK-D2 here in the wild type, but not in the mutant. And that's a readout of Fanconi mutant biology. Appropriately, the wild type gene corrects the mitomycin C sensitivity here, but the mutant allele does not. And you can notice here that the mutant cells are WEE1 sensitive, WEE1 inhibitor sensitive, while the wild type cells are relatively resistant. And all of these cells have equivalent PARP inhibitor profiles. And finally, we've actually developed some murine lymphomas from a FANK-D2 knockout mouse, which we're actually using as a tumor model in the lab. And we have a het lymphoma versus a knockout lymphoma. And we can actually show there's absolutely no effect of WEE1 inhibitor on the hets, but the WEE1s show some retardation of growth in as a single agent. So these are just the beginning of experiments that we're doing to try to lay the basis for using this eventually in patients who actually have mutations in these particular genes. So our pipeline is verifying these pathogenic mutations from sequence data, looking at tumors obtained from knockout mice, FANK-D2, as well as FANK-C, and utilizing these synthetic lethal data to try to support their use eventually in some kind of clinical trial. So with that, this is just all the people in the lab, both present and past, who contributed to this work, and just a partial list of all the various people we work with here at Yale. With that, happy to take any questions.
Questions for Gary?
You always have different colored bowling balls?
It's a good way to get out of work.
These adults that you're finding with the FANC mutations in their tumors, are they homozygous? Are they genomic? Are they just unrecognized FANC patients?
No.
These are just acquired.
So the first case I told you was a patient with a germline mutation
Right.
in one allele, and the second allele was acquired.
Right.
The second case, both alleles were actually acquired. So it's a mixture, almost certainly.
Do you approach your patients who are developing leukemia, other tumors in the kids differently? I know that for transplants, they have to have reduced doses, for example. At least that they used to.
The overarching motivation is really to get patients transplanted early, but not too early where you create such morbidity from the transplant. So, and so the doses are markedly reduced, as you're pointing out. They've also included agents like fludarabine, which have remarkably low toxicity in patients with, and that's when fludarabine came on the scene, patients with Fanconi who were getting transplanted went from minority surviving to 3/4, 80% long-term survival. But of course, it's both a good problem to have, I guess, the fact that they grow up and now we're getting a whole host of solid tumors. The sweet spot is like between 5 and 10. Yeah, so I would say that the vast majority will not get leukemia till after the age of 10. So when we start seeing their counts kind of drifting down, assuming then there's going to be an upward pressure on their bone marrow to keep up, that's kind of when we try to get them transplanted.
Do you have any sense for what percent of Fanconi's, obviously it's kind of a impossible question, I guess, are not recognized when we see these young adults with the bone marrow failure states, and then we screen and they turn out to have Fanconi's?
I think the percent is hard to say, but there's definitely patients there. I get called over by the head and neck team a couple of times a year. No, and there's clearly about a third of Fanconi patients do not look like the textbook. And so, yeah, I get called over by head and neck team, and usually to see a patient who has had inordinate amount of toxicity from the chemo. And so we identified a 50-year-old a few months ago. With further discussion, turned out had a family history that he was the product of a first cousin marriage. So that actually didn't take too long to tease out.
Good. What you do see is that the FANC genes are among the most polymorphic genes that we see. The amount of variability in the population is just enormous. So you may have things that are kind of
Borderline polymorphism.
Yeah.
I mean, borderline biology.
Well, you know,
Less penetrant.
We also don't even know. I mean, most of these Fanconi genes have no identifiable functional motifs. We don't really know their specific function. So it's a little hard to know, you know, to make predictions based on that.
Do the knockout mice have defects?
They have surprisingly few defects. They do not have spontaneous bone marrow failure. They don't get spontaneous cancer. The tumor model I showed you is P53 het background, so you need that in order to get them to elaborate any cancers.
No skeletal defects.
I'm sorry?
No skeleton.
No, sorry.
Gary, thank you.